Enzymes are made of proteins, and proteins are made of long chains of amino acids, and these get folded into specific shapes to allow the enzyme to perform its role within the cell. Every enzyme has this very specific active site. This is the site that catalyzes the reaction. The active site has that specific shape for each substrate. Each substrate, such as starch, has a very specific shape, and only the enzyme that breaks down starch, amylase, has an active site that will fit starch into it. And so enzymes usually only catalyze one reaction. This simplified model is called the lock and key model of enzyme action. The enzyme is like the lock with a specific shape for the substrate that's like the key. The enzyme is not rigid in its structure, however, and changes shape a little as the substrate is attached. So the enzyme closes around the substrate to make it a tighter fit, and this more complex model is called the induced fit model. As enzymes are held together by bonds, changes to their environmental conditions can break these bonds, and therefore change the shape of the active site. This changing of the active site denatures the enzyme, and they stop working. Two factors that can change the active site shape of an enzyme are temperature and pH. When we increase the temperature of a substance, we increase the kinetic energy. This kinetic energy increases the number of successful collisions between the enzyme and the substrate. Up to a point, the enzyme has an optimum temperature at which it will work. After that, the increased temperature breaks the bonds that hold the enzyme into its specific shape and changing the shape of that active site. This is called denaturing. Remember that enzymes are not denatured at low temperatures but have that less kinetic energy.